Good morning. I would like to thank the organizers, first of all, for giving me this opportunity to talk here. Slightly different from what I gave in the title, I'm going to say the story which is related to chromatin is what I'm going to present today, where some stochastic thermodynamics, how we can use these ideas from stochastic thermodynamics to understand certain biological system, here chromatin. So my title is Stochasticity, Nucleosome Positioning, Chromatin States, and Gene Regulation. As I go, I'll explain what these terms mean. So as we all know, DNA is a very long polymer, and it is a polymer made of ATGC, nucleic acids, and it codes for all the cellular processes that is going on in our cells. So we have this long codes, and it is a long molecule inside our nucleus. And we all know that for every process that is happening in our body, the DNA is being read. So interestingly, it's not just read, the cell also needs to prevent it unwanted reading. What do we mean by unwanted reading? If you think about it, as we speak, many of our cells, the DNA is being read. For example, if we take food, the DNA in various cells will be read to do the metabolic activities. If we have a wound, for that wound to be healing, the DNA in those cells has to be read, and the proteins, the material needs to be produced to heal that wound. But when you don't have a wound, you don't want those cells to produce those material, otherwise it would lead to some disease. So you want DNA reading in a regulated manner. You don't want unwanted reading. Therefore, you want to have a control over this reading of this genetic material. To do that, DNA is nicely packaged and folded. DNA is bound with large number of proteins in the cell. It's not a bare polymer inside the cell. It is covered by large number of proteins, and some cartoons are showing here and some images. And this covered folded DNA is called chromatin. So this is a cartoon of this DNA packaging, which is supposedly in different levels. So if you look into the details, this is the typical textbook picture, which is partly not fully correct. But however, this is what is being taught in the textbooks, that DNA, of course, we know about DNA, which is double helix, which is wrapped around this yellow ball-like things to form an array of nucleosomes. So this is an array of nucleosomes where you have many nucleosomes like beads on a string. And they are supposedly further packaged. That is what the textbooks say. However, recent results do not support many of this cartoons in the textbook. And finally, it has some three-dimensional organization inside the cell. We know very little about the higher order organization, but recent experiments are trying to tell us, or they are exploring, or they are probing what is the higher order 3D organization. But what is known is that the DNA is actively organized in 3D. It's not in thermodynamic equilibrium. They are organized and maintained in a three-dimensional structure actively by machines that use ATP from the food we eat, essentially. So what is the organization of the genome in the living cells? It's an open question. Part of it we know, part of it we don't know. And somewhat like this cartoon would represent a kind of a heteropolymer organization, where you have a long polymer having many proteins bound. And the prominent proteins are this called nucleosomes. So these blue things are nucleosomes. So there is this blue proteins around which DNA is wrapped, about which I will talk a lot about. And there are many other factors, and there are many other chemical flags, signals, chemical modifications, and all together, DNA is packaged into a three-dimensional organization. But what is interesting is that we have different cells. We have cells in our skin, we have cells in our eye, cells in our heart. They are very different cells, but they have exactly the same DNA. We all started from single cell and divided. So all our cells have exactly the same DNA, but these cells behave very differently. So it's an interesting question that how these different cells which have exactly the same DNA, but they have very different behavior. It's like you have the same program, but it runs differently in different parts, whether you run in Bangalore or somewhere in New York. If the program runs differently. It's that kind of puzzle, right? So, but what is known now is that the function of a cell is not decided by the DNA sequence alone, also decided by the states of the chromatin. So chromatin is what is the biologically relevant state of the polymer, and depending on different states, microstates and macrostates of chromatin, one would have a different behavior or fate of each cell. Now, what are these different states? What are the microstates? These are interesting questions, and we are researchers on to understand different states of chromatin and what are the relevant states, what are the states that is biologically relevant. So, how different states is actively maintained and what are the states are interesting questions. And I would try to tell about some research that we did in trying to understand some of the states of chromatin in some very specific context in biology. But interestingly, this states is actively maintained by ATP-dependent chromatin remodeling machine, and this is crucial. Now, if you go a little bit into the details, so if you go and look at some part of the cell, like if you look at this part of some part of the chromatin, if you look at this, focus on this part, it is like essentially like a is this like a beads on a string? So you have nucleosomes, an array of nucleosomes, as I said. So this is the first level of packaging. You can think of this as some particles on a one-dimensional line. So you have DNA, which is like a one-dimensional track on which you have many particles bound. This is good for any simplest picture what any physicist would imagine. And one of the important interesting question is there a pattern in this organization of this ball-like proteins, this whole thing called this nucleosomes? So is there a pattern in the organization of nucleosomes? That is an important question. To understand that, let us understand a little bit more about nucleosomes. So nucleosomes are these yellow proteins on which DNA is wrapped. So DNA can wrap on this protein, and due to electrostatic interactions, they form this and then they wrap like this. So if you think of this as a chemical reaction, this plus this forming this, the delta G is hugely negative, minus 40 kBt, or of the order of that, highly negative, it's a large negative number. So what does it mean? It means that forming a nucleosome, that is DNA wrapping around this protein called histone, is energetically favorable. So this state is energetically highly favorable. So nucleosomes state is a highly stable state. So what does this suggest? It means that any thermal dissociation will be nearly impossible, at least in the time scale of a life of a cell, or the time scale that is relevant in biology. The dissociation thermally will be nearly impossible, and it implies that we need extra machinery to disassemble or to reorganize. If you want to move around these proteins, we need extra machinery which would use energy from outside. Thermal energy cannot do this job. And biologists have discovered such machines. There are different classes of machines. They do sliding. For example, this cartoon from Albert's book shows that this machine can slide this nucleosome from here to here. Look at this white patch. It was pretty close to the nucleosome, and in this white patch is far away from the nucleosome, which means the nucleosome got repositioned. And in this process, they use ATP. So there is ATP-dependent sliding, and there are many enzymes that do that. There is ATP-dependent disassembly also. So these machines really catch hold of this nucleosomes and eject it. So they can do this. So this kind of machines are implicated in disassembling. So what inside is a DNA-wrapped nucleosome, and this is the machine which literally holds it and plucks out the nucleosome. So biologists know about the ATP-dependent machines that use ATP and either reposition, that is slide, or disassemble nucleosomes. So now let us come back to this question. How are these nucleosomes, which you can think of as sterically interacting particles, organized inside the cells? Are they randomly organized, or is there a pattern? That is the question, as a physicist, one would be interested. What experiments showed is that there is a non-random organization of nucleosomes. So I quote non-random because it's, you will see that this is the word used. So there is some kind of regular pattern of nucleosomes. What experiments found is something like this. So this, what is plotted here is nucleosome occupancy. What does it nucleosome occupancy means? If you think this black line as the DNA, nucleosome occupancy is the probability that a DNA site is covered by a nucleosome. So if you, experiments can measure this steady state probabilities that a particular site on the DNA is covered by a nucleosome. So if you compute this, or if you measure this experimentally, there are methods to measure this, then in steady state one sees this pattern if you look at gene regions. So those regions, you look at those regions in the chromatin, which is coded for proteins, which is the genes. And if you look at the nucleosome occupancy there, they have a pattern which is like this. So to explain this pattern, this is known for some time. Roger Kornberg and Stryer wrote a very famous paper many years ago, 1988. The title of the paper is Statistical Distribution of Nucleosomes, Non-Random Locations by a Stochastic Mechanism. So essentially, what Kornberg and Stryer argued is, in a physicist language, it is the following. They argued that if you imagine nucleosomes which are dynamic on a line, it is essentially like a Tonks gas. So you have a one-dimensional line on which you have a sterically interacting particles like hardcore fluid, and they could have various different arrangements, and they are always, if they are near a barrier, so which is like a boundary or a wall. So if you have a Tonks gas or a 1D fluid near a boundary, and if you look at the G of R, you would look, it would look like this. So this is essentially the argument that Kornberg and Stryer made, which give rise to this kind of pattern. So this would, so what is important here is this barrier, which is some other proteins, which is relatively much stable and bound there, like, and it acts like a wall, and near a wall you will get this nucleosome occupancy, which has this behavior, which is like G of R for a liquid or a fluid. After that, recently there's a lot of new experiments in trying to understand this, and recently in Science in 2011 from Frank Pugh lab, they showed that this kind of oscillatory this kind of G of R like organization is only possible if you have ATP. So they did experiments in cell extract with and without ATP. So if you do not have ATP, you have nearly flat line, and if you have ATP, only then you get this pattern, which is basically the signature of a fluid or fluidity essentially. So ATP-dependent activity is necessary for maintaining the nucleosome positioning, which is relevant in cells. Then there are many other experiments recently studying the nucleosome organization. Another very interesting set of experiments by Small et al. From Northwestern and Brown et al. From California. They did single cell experiments. That is, they looked at each cell by cell. And what they looked at nucleosome organization at a particular region. It's called promoter. The names doesn't matter. Names don't matter. But in an important region called promoters of cells, when they looked at the nucleosome organization, what they found is that different cells show different organization, slightly different organization. And there is, some cells have some this organization, some cells have this organization. So all of these have like 3 nucleosomes, or some of them are 2 nucleosomes. Some of them are differently positioned. This region, the violet region, which is a particular sequence called TATA, and this sequence is very crucial for reading the gene. Many of the reading machineries bind here. So this, in some cells, this region is open. In some cells, these machines are covered. These regions are covered, and these 2 other colored green and blue, these are also some 2 protein binding sites. Those are also crucial, and they are in some cells they are covered, some they are open. So under the same condition, different cells show different nucleosome positioning, which means there is stochasticity in nucleosome organization. And they quantified this. For example, the various experiments where they could come up with 8 states. So this is a state where all the 3 nucleosomes, if you take a small region, there are 3 nucleosomes. Sometimes there are 2 nucleosomes, but arranged in 3 different ways. Sometimes there are only one nucleosome and arranged in 3 different ways, and there is no nucleosome. So they measured the probability of this 8 kind of coarse-grained macro states from this experiment. So in this context, we started asking various questions, theoretically doing from a theoretical perspective. So the question we started asking is, given that nucleosomes are dynamic, they can slide, there are always remodeling machines, so they can slide and disassemble, they can bind, dissociate, and slide. What must be the underlying dynamical rules to obtain the experimentally known pattern? What is the role of ATP-dependent chromatin remodeling in maintaining this experimentally seen, observed chromatin organization? How nucleosome organization would influence the 3-dimensional looping and organization of chromatin? How certain non-nucleosomal proteins might affect the chromatin organization? So these are the questions that we started asking. So the first part, we would think of the one-dimensional picture where we will study the nucleosome positioning along the DNA, which is a 1D track. So we started with a simple model where nucleosomes can bind, slide, and dissociate along a track. So we started with simple minimal models and added details. We added details like, first we started with simple model and then later added details like affinity with respect to sequence and so on and so forth, and the barrier which would prevent sliding of nucleosomes from one location to the other at a certain locations, and so on and so forth. So the rates, the details of the model is in deciding all these rates. So the R on would depend on the available space because they are sterically interacting. R off would depend on the intrinsic rate and some sequence affinity VI, which we can get from some bioinformatics information. And VA is the active disassembly, the energy used, like the delta mu. And there is a sliding which is essentially 0 without ATP in the time scale that we are interested in. And otherwise, there is a sliding. So, and we played around with these details of the models. And we essentially did kinetic Monte Carlo simulations. And so at some, wherever we can, we also did some analytical calculations. So if we first ask, what would happen if you switch off all the ATP-dependent moves in our simulation, you would not get this positioning, essentially because this is essentially frozen. There is no dynamics. So these are like randomly frozen nucleosomes, and if you average over them, you'd get a flat line, while if you have some entropy and movement, then you would get this oscillatory positioning. So one of the student, PhD student, Hongyo Hariri, he started thinking about what could be the underlying kinetics that will lead to the experimentally seen nucleosome organization. So we have this nucleosome organization from different experimentals by Brown et al. And Small et al. So this is from Small et al. So to obtain this kind of pattern, what should be the rules? If we just have this on, off, and sliding in a sequence-dependent manner, will we get it? Or do we need something more? And I am cutting down long story just to say the final message. The message is that it is not enough to have this on, off, and sliding dynamics. We need to go beyond the nucleosome kinetics. We need to add certain events to get those specific experimental details. So binding of certain extra proteins at some locations is needed. Only then you would get this. And so that this prevent this nucleosome, not only this prevent this nucleosome from sliding and occupying the space, these proteins have to remove the nearby nucleosome by recruiting some remodeling machines. So there are some other biochemical evidence that certain proteins binding on certain regions on the DNA can recruit remodeling machines locally and disassemble the nucleosome. So it turns out that nucleosome disassembly by this transcription factors, as TF, and their name is FOFO. Here doesn't matter the name, but it is, we learned that without having this non-equilibrium disassembly of nucleosomes, one will not obtain the experimentally seen organization. So we could, with our with two parameters. One is the rate of affinity of this as one parameter, and the disassembly of this due to this as another parameter. All other parameters are earlier decided from various in vitro experiments. We could obtain this organization of nucleosomes in off state and on state. So this is the off state. You can see that this data, this dots and circles and squares are experimental data from Brown et al. And this is our simulation results. In the off state, most often you see that all the nucleosomes are present, and rarely you get all empty, but very often you get sites with one nucleosomes. This is not zero. And in the on state, it is populated mostly by empty sites, but however, this kind of sites are also possible. So there is a distribution, and there is some probability of having different states such that the gene is on and off. So so this is like a promoter region switching from off to on state because of nucleosome dynamics and protein dynamics. And once they are in the on state, it can produce mRNA, which is the gene expression. And one can look at the distribution of mRNA. So if you look at the distribution of mRNA, one would find various interesting distribution. When it is off, it will be like a distribution like the red curve. When it is on, it will be more like this blue curve. And for some parameters, you could get the bimodal distribution, which is biologically very interesting because a population of cells is producing the protein and another population of the cell is not producing this protein, and this can have interesting ramifications as far as the fate of that cell is concerned. Now we wanted to understand this is more in detail, where given a nucleosome dynamics, we can compute how long a particular region of interest will remain open, exposed, before it getting covered by another nucleosome. So this is, let us say, some region of our interest, this region M, and there are many nucleosomes that do bind, dissociate, and so on and so forth. And you have a highly stable barrier here, which is permanently fixed there. So near this barrier, you have a location which is of interest, let us say it's Tata box, or the TA sequence where some of the protein has to come and bind. If you have such a scenario, and given that nucleosomes is dynamic with some rates, how long this region will remain open. So this is some work we did with my student Josna Parmar and my colleague Dibyendu Das in physics. We could compute the first passage time of covering this patch of size M. So you have a patch of size M. What is the mean first passage time that this site will be covered if you start with an open state? So you start with some state from a condition such that the first nucleosome is L away and the patch is L1 away from the barrier. And if you start with such a thing, one can write down mean field, one can write down this first passage time equations and compute in a limit whenever binding dissociation dominates over sliding. So we neglect sliding in these calculations and we can compute it, and it matches quite well with experiments, with simulations. What is interesting is that if you think of, one would naively think that if this comes off and binds here, this will be covered. So one would naively think that this is dominated by on rate or off rate, those time scales. So it turns out that this is the 1/k of time scales. So this is not like just one time scale. It is a combination of many time scales. So there are multi-particle dynamics dominating this time scale to keep this open. That is what the take-home message is. And we could do this now incorporating sequence, where you would get sequence-dependent time for that to be remain open. We can also ask the question that given a particular promoter which has a particular architecture, what do I mean by promoter architecture? There are some binding locations for those proteins. There are some binding locations for some of the proteins, if you know that they are L1 distance apart, for different genes, there is different architecture. And given this promoter architecture, this time scale would differ. So how this time scale depends on the architecture of the promoter is something which we can explore. And this is how TL, the first passage time, depends on this L and L1 is for different L1 values. How would it change is something that we also computed. Now, as I said, many of the experiments measure basically the occupancy. Occupancy means in steady state, what is the probability that a particular region is covered by a nucleosome? And biologically, what is relevant is that some region of interest, the pink region, is covered by a nucleosome, but eventually it needs to be bound by another protein. Only then some biological activity will happen. So if you ask this question, how long it takes for this protein to bind here, what is the time scale of this transcription factor to bind here? If it was naked and bare and available, it would bind with some rate Rp. And K plus and K minus are the nucleosome binding dissociation time scale. If this was bare, the time scale would be 1 over Rp. But since there are nucleosomes and nucleosome dynamics is going on, the time scale will be different. And if you look at the time distribution of the time scale, how long it takes for this to bind here, we have 3 different cases for one value of Rp, which is the 15 second is the time scale. What we find is that in these 3 cases, the steady state occupancy will be exactly the same. So even if steady state occupancy is exactly the same, the distribution of the time scale, the time needed for it to bind, would be very different. And this is interesting because biologically what is relevant is this time scale for this transcription factor to bind, not really the occupancy. Therefore, even though experiments would measure the same occupancy, there could be very different dynamics, and this would depend on the stochastic dynamics of nucleosomes. So same nucleosome occupancy, you can have very different binding times. So in the second part, we studied how do nucleosomes influence the 3-dimensional organization of chromatin. So this is, so far we looked at the 1-dimensional organization of nucleosomes, and chromatin is actually a 3D polymer, a polymer in 3D. Therefore, it is a question to ask that how do nucleosomes influence the 3-dimensional organization of chromatin. This was done by a student Gaurav. So let me just go slightly, sorry. I'll just, since there is time, I'll just tell you a little bit more detail. So what is known in the current literature, what is known is that nucleosomes bind, and when this DNA wrap around these nucleosomes, there is an entry and exit. The entering and exiting parts of the DNA would make some angle. So this is the textbooks would show you such organization of nucleosomes, such organization of the chromatin in, that's what one would expect. And a lot of, so the physics of this zigzag is that you have a DNA which is entering and exiting the nucleosome, making an angle. So there is an entry-exit angle, and there is a stiffness for the linger region. So this length is much smaller than the persistence length. So this length is about 20, 30, 40 base pair, which is much smaller than 150 base pair, which is the persistence length of the DNA. Therefore, this would be nearly stiff. So since this is nearly stiff, and given that they are periodically organized, one would naively assume such zigzag-like structure for nucleosomes. So that is the picture that is in the textbook. But many recent experiments found that in vivo, the experiment, so this is called a 30 nanometer structure because this is a width of 30 nanometers. So this has a width of 10 nanometer, another 10 nanometer and 10 nanometer in between. So there is a 30 nanometer width. So in literature, this is called a 30 nanometer structure. So what various experiments found is that in vivo, there is no 30 nanometer structure. But in vitro, you can see this 30 nanometer structure. if you can reconstitute this in vitro and do X-ray crystallography, and you would see nice 30 nanometer structures, but in vivo, if you look at it, you would not find this 30 nanometer structures. So we looked at these models for chromatin and that predict various organization and structure, and we found that the models so far only consider this nucleosomes, histones, that too in a regular organization of histones. And once you consider that, you will have immediately this kind of zigzag-like picture. But in reality, what we know is that there are many other proteins. They can bind along the linker region and bend the DNA. So no model so far has accounted for the role of DNA bending known histone proteins. There are proteins like HMG which would bend the DNA. What would be the chromatin organization in the length scale of a gene given a concentration of DNA bending proteins and a given nucleosome organization? So this is the question that we asked. If you have DNA bending proteins apart from nucleosomes, what would be the organization? Will this zigzag picture persist? So to do that, we did two kinds of simulations. One, we did a simple freely rotating chain model simulation, and we also did detailed Brownian dynamics simulations. So in the Brownian dynamics, we took bead spring models with nucleosomes that would bend the DNA with an angle, and you have other proteins which would bind and bend. In the freely rotating chain, DNA is considered as a semi-flexible polymer. So in the freely rotating chain, semi-flexible polymer, semi-flexibility is essentially decided by this angle theta, and phi is free to rotate. So that is the freely rotating chain. And wherever nucleosome is bound, it will make an angle theta n, which is the nucleosome angle. And wherever a protein is bound, it could make a different angle theta p. And in this model, if you have no known histone proteins and regular arrangement of nucleosomes, one would find our simulations and would find regular arrangement like this. But if you add known histone proteins, the proteins that bind and bend the DNA randomly in the linker regions, one would find irregular structure. So you might have seen a 1 week ago, there is a lot of experimental, there is a science paper and a lot of news about how chromatin is irregular. So there is experiments kind of indicate that chromatin is actually irregular. There is no regular organization beyond the organization of a nucleosome. Therefore, what we suggest, we could hypothesize the underlying reason for such irregular nature that could be the proteins that bind and bend the DNA. So under what conditions will one observe irregular chromatin, and under what conditions one will observe regular structure? This is interesting questions. So we have various parameters. So we could vary two parameters which are important. One is the linker length. Linker length is the length between two parts of the, so this is the length between two nucleosomes. So this length is one parameter that we can vary. So if we vary linker length, and another parameter we can vary is the protein density, the known nucleosomal proteins which would bend the DNA. We could ask the question, what is the density of those proteins? And we could look at, for these two parameters, if you have large linker length and very large protein density, one would find irregular organization. While if you have very small density of known histone proteins and some particular linker organization which is small because it will not bend, then you will get zigzag-like structure. And you could also vary the bending angle of these proteins. So this is the bending angle and protein density. So if the bending angle is very small, then again you will get zigzag for a given protein density. So there is a quote-unquote critical protein density above which it would become irregular for any bending angle. Similarly, for any linker length, there is some protein density beyond which it would become irregular. So this is the prediction of our theory. And what is the density of known histone proteins beyond which the zigzag would disappear? This is an interesting question. Is that density biologically relevant? So if you look at so what is experimentally measurable is how what is the probability that two nucleosomes will come in contact? So IK is the probability that kth nucleosome will come in contact. You could measure that experimentally. And what experiments found, so typically in a zigzag structure, one would expect that when k is equal to 2, that is next neighbor nucleosomes would come in contact, that will have high probability. So therefore, if you have next neighbor coming in contact, there will be a peak at k equal to 2. That's what one would expect. So this is the blue one. When there is no known nucleosomal proteins, you would have a peak at 2. And as you add known nucleosomal proteins, which would bend the DNA randomly, then this peak would come down. And for some density, this peak at 1 and peak at 2 will be equal. And at that density, we define that at that density, zigzag would cease to appear. So that density is turned out to be around 30% when I1 is equal to I2. So this is the density of known histone proteins where I1 is equal to I2, where zigzag cease to appear. And that is neighbor and next neighbor nucleosomes interact. And this is comparable with recent micro-C experiments from one from a lab. And under what conditions one will observe irregular chromatin? As I said, one can study this. So, and one can get details, understanding of how this happens. And interestingly, this 30% that I talked about is biologically relevant. It is known that these proteins, called HMG proteins, they are highly abundant. So they are as abundant as 50%. So there are much more non-histone proteins that bend DNA available in cells. So therefore, this density is indeed biologically relevant. So to summarize what we have been doing, we have been studying physical models for nucleosome positioning. We have been studying dynamics of nucleosomes and how nucleosome positioning will affect 3D organization, and the DNA bending proteins will influence 3D organization of the chromatin. So finally, I would acknowledge my collaborators. So this work is done by 3 graduate students. Josna, who did the first passage time calculations, Hungyo did that early work on 4.4, and Gaurav, who did the 3D organization part. This is in collaboration with Professor Dibyendu Das, who is a colleague of mine in the physics department. Early part was done in collaboration with John Marco, who's in Northwestern University. And the last part on 3D organization was done in collaboration with Mandar, and one part with one of our experimental colleague, Professor Bhatt, in the biosciences department. So funding is thanks to DBT and CSIR, and I'll be happy to take questions.
The last part that you showed, are these equilibrium simulations, or do you have ATP activity?
No, it's essentially equilibrium. There's no activity there. The only activity would come is that this organization of this nucleosomes would, at the moment we have taken a given organization, what is the 3D, but this nucleosome organization could be, will change in a non-equilibrium manner, in the sense that ATP-dependent machines could reorganize these nucleosomes.
No, the polymer dynamics is completely...
This is equilibrium only, this, yeah.
I had this questions. You talked about the linear length and the the angle between the...
Yes.
Right? But
yes.
The number of turns, is it always like 2 into 360 degree, or will there be a difference in that as well?
Let me see. In this picture, you're asking.
So is it always like to complete?
Yes, it's always 1.7 turns. Like it's like not full 2, like 1 point. Yeah, it is always. Okay, so it is possible. Some fluctuations around is possible, but that is if you look at crystal structure, that's how you would see. Many people, many labs have crystallized this and done X-ray crystallography of nucleosome structure, and they all see this. But it is also known that they can there will be some fluctuation, yes, but predominantly it will be an angle of 60 degree, and even in this model, there will be some fluctuation around this angle that one fixes, because these are thermal simulations, right?